Aloha, everyone. Thank you so much for joining us today to learn about nutrition from an epigenetic perspective to prevent and mitigate autism and ADHD. My name is Dr. Renee Dufault. I am a retired public health service officer, and this presentation is sponsored by the Food Ingredient and Health Research Institute. Today we're going to talk about a tested and proven evidence-based model that explains why autism and ADHD occur so often in the United States and what parents can do to prevent and mitigate these debilitating conditions in our children so they will have better health and learning outcomes. First, let's provide some background. The Food Ingredient and Health Research Institute is a 501c3 nonprofit organization. We achieved that status in 2010, but we were founded in 2009. Our organization has numerous publications now indexed on PubMed. We have published important findings. For example, we now know that dietary mercury and lead exposures lead to the development of type 2 diabetes and autism. And we know that dietary lead exposures lead to the development of ADHD. When I worked at the United States Food and Drug Administration, I sent a field investigator out to high fructose corn syrup manufacturing plants to collect samples for mercury analysis in 2005. My collaborators and I were only allowed to publish our findings of mercury in high fructose corn syrup after I retired in 2008. We published our findings in the Environmental Health Journal in 2009. Since that initial publication, inorganic mercury residues have consistently been found in some corn sweeteners by Canadian and other researchers. It is important to understand that heavy metals are found in many different ingredients throughout the processed food supply. Mercury and lead residues are found in many processed foods because the law allows these heavy metals to occur in specific food ingredients. After we published our findings of mercury in high fructose corn syrup, we published our first model explaining how mercury in high fructose corn syrup can impact the way genes behave in our children. We show how mercury, together with high fructose corn syrup, can create mineral imbalances inside the body and conditions for the development of autism and ADHD. That first model now has 118 citations and is being used all over the world to conduct research on children with autism and ADHD. It was published in the Behavioral and Brain Functions Journal entitled Mercury Exposure, Nutritional Deficiencies, and Metabolic Disruptions May Affect Learning in Children. Here we are 12 years later, and the world is finally paying attention to the heavy metal contamination problems in our food supply. On July 27, 2021 this year, the WHO, the World Health Organization, announced it wants data on heavy metals in food. This is a screenshot of the article published in the Food Safety News on July 27, 2021. Why now? Why all the attention now on the allowable heavy metals in the food supply? I'd like to think it's because we published our third and final evidence-based model on July 20th, 2021, this last summer. This article explains how dietary mercury and lead exposures lead to the development of autism and ADHD. 7 days after our model came out, our new model, WHO asked the world for heavy metal data in food. And 2 months later, the United States Congress issued their report on heavy metals in baby food. The title of the U.S. Congressional Report, published on September 29, 2021, is New Disclosures Show Dangerous Levels of Toxic Heavy Metals in Even More Baby Foods. There is nothing new, of course, about this problem of toxic metals in the food supply, except that our recent model is backed by numerous clinical trial data from studies conducted all over the world. Let's get to the essential questions we face today. Why is it that Western diet, also known as the SAD, the Standard American Diet, leads to increasing autism and ADHD prevalence. How has food changed over the years? How are we as organisms in our genes changing in response? The answer to these essential questions, in order to answer them, we must take a hard look at how food has changed over the years and how our genes are responding to the new foods we are eating. The answers lie in epigenetics. Epigenetics is the study of gene-environment interactions, how your genes behave in response to the changing environment. What we see here are plant genes responding to their environment. What is the change you see between the two pictures? The lack of water impacts plant growth, just as the lack of proper nutrition impacts child growth and development. Epigenetics is a study of changes in organisms that are caused by changes in gene expression rather than changes in the genetic code itself. Genes can express themselves by turning on or turning off in response to their environment. You have the same set of genes you were born with, but they turn on and off depending on the environment in your body. What we eat determines the way our genes behave. If our diet changes, our genes change their behaviors in response. This is a picture of the PON1 gene. When it isn't turning on, you will not be able to produce the enzyme your body needs to break down the pesticide residues commonly found in the American food supply. PON1 gene expression requires adequate dietary calcium intake, and it also requires a clean diet free of heavy metals. Heavy metals can suppress or dampen this gene's activity. This suppression means the gene won't be able to produce enough of the enzyme you need to break down the pesticide residues that you eat. These are the epigenetic risk factors for poor birth outcomes related to poor diet. Heavy metal and pesticide exposures via maternal diet, gestational type 2 diabetes, maternal obesity, and low 1 PON1 gene activity. It is important to understand that after a child is born, the development of autism and ADHD continues to be related to heavy metal and pesticide exposures from the food the child eats. Their gene behavior and metabolism is set up at birth. An obese mother will set her child up with the metabolism to become obese. She can also pass heavy metals on to her baby via cord blood. What is the impact of heavy metals on child health? Heavy metal exposure. Heavy metal exposures are associated with autism, ADHD, and developmental delay. Children with autism always have elevated mercury and lead exposures. We know this now because so many clinical trials have been done all over the world. Children with ADHD always have elevated lead exposures. Exposure occurs via ingestion, primarily from processed food consumption. There are allowable levels of cadmium, mercury, lead, and arsenic in specific food ingredients found in highly processed food. In addition to processed food intake, these exposures also occur via ingestion of paint chips, contaminated soil and water, and inhalation of contaminated air. But the truth is that the most common exposures come from food, processed food. There are allowable levels of cadmium, mercury, lead, and arsenic in specific food ingredients found in highly processed foods. These exposures must be minimized in all children to improve their behavior and learning outcomes. Parents can reduce their child's exposure to toxic heavy metals by simply making changes to the family diet. If the exposures are not minimized, then heavy metals can build up in the child's body tissues, including the brain. The heavy metals can also suppress the PON1 gene activity, making dietary pesticide exposures more toxic. This can impact learning and the capacity to learn, as well as lead to the development of chronic diseases, such as type 2 diabetes, heart disease, Alzheimer's, and Parkinson's. What you feed your child today will determine his or her health outcomes tomorrow. Health education programs must teach young people to avoid these exposures because they are the parents and leaders of tomorrow. These are allowable heavy metal impurities in many food ingredients. These are some of them. There are allowable heavy metal impurities in many food ingredients. Here is a chart of the food ingredients with GRAS status, which means they are generally recognized as safe by the FDA and can be used by food manufacturers without restriction. This means manufacturers can add as much of these ingredients to their products as they want to. In addition to these food ingredients, there are other food ingredients with allowable heavy metal impurities that may be used by food manufacturers under certain circumstances. Parents need to know which food ingredients to avoid when grocery shopping. For example, you'd want to avoid caramel as a food ingredient because it has allowable 3 parts per million arsenic, 10 parts per million lead, and up to 0.1 parts per million mercury. Another one that you'd want to avoid is sodium benzoate with its allowable 2 parts per million lead and 1 part per million mercury. Mercury, lead, and arsenic residues are found in many processed foods because the law allows these heavy metals to occur in specific food ingredients. These are examples of restricted use food ingredients. Blue 1, blue 2, yellow 5, yellow 6, red 40. As you can see, there's up to 50 parts per million allowable chromium in blue 1, up to 10 parts per million lead in blue 1, blue 2, yellow 5, yellow 6, and red 40. And there's up to 1 part per million mercury in blue 2, yellow 5, yellow 6. You get the picture. This is what's allowed by the regulations. This means that with restricted use ingredients like these, the food manufacturers can add only so much of these ingredients to their food products. It's not, they have limits. These are just some of the food ingredients that parents need to avoid as they grocery shop. Food color consumption has been found to impact child behavior, leading to problems such as hyperactivity and inattention. In the United Kingdom and Europe, food products containing some of these ingredients must be labeled with a warning on the packaging. This labeling requirement has been in effect since 2010. So the warning label used in the United Kingdom and the European Union must say, may have an adverse effect on activity and attention in children. And this warning is required in the UK and the EU when using yellow 5, yellow 6, and red 40, and several other food ingredients. In 2018, we did an analysis of comparable data sets available in the United States and the United Kingdom since and before the food labeling requirements took effect, and found a tremendous difference in the prevalence of autism and ADHD between these two countries. We found that food labeling requirements may explain the lower ADHD prevalence in the United Kingdom. How big is the difference in prevalence between these two countries? In 2016, the percent of children diagnosed with ADHD in the US was 9.4%, and the percent of children diagnosed with ADHD in the UK was only 1.5%. So the food labels went into effect in 2010, and 6 years later, then we see this difference in the prevalence. Today, CDC is saying 16% of all children in the U.S. are ADHD, so it's been jumping up higher all the time. The prevalence of autism among 8-year-old boys between 2004 and 2010, before the food labeling requirement in the UK, was 11 out of 1,000 in the U.S. children and 4 out of 1,000 in the United Kingdom's children. So this prevalence was occurring in autism before the food labeling went in effect. So what could cause this lower autism in the UK? It's clearly not related to the food color warning labels because there was a huge difference in the autism prevalence before the warning label took effect. The warning label requirement only began in 2010. Why is the autism lower in the UK? What is the epigenetic risk factor responsible for the higher autism in the US? In 2012, my colleagues and I published the second model to further explain how some of the epigenetic risk factors lead to the development of poor outcomes in the United States. We compared food intake patterns in the US to Italy because we found a difference in the PON1 gene activity between people in Italy and people in the US. Here we see how exposures to heavy metals and high fructose corn syrup in the US can lead to disruptions in the production of key proteins needed for normal heavy metal and pesticide residue excretion. High fructose corn syrup consumption in Italy does not occur due to trade restrictions. High fructose corn syrup consumption in the U.S. leads to dietary calcium and zinc mineral losses, which impact heavy metal and pesticide excretion mechanisms. This second model updated our knowledge on the mineral imbalances that occur with the consumption of high fructose corn syrup and the impact of these imbalances on gene behavior. With the calcium losses, PON1 gene activity decreases, and pesticide residues are not excreted from the body. High fructose corn syrup suppresses PON1 gene activity. With the zinc losses that occur from consuming high fructose corn syrup, the metal carrier proteins are not produced in adequate amounts to allow the body to excrete mercury and lead residues that we get from the food supply. High fructose corn syrup consumption does not occur in the United Kingdom, Italy, or other European countries. Thus, there is less autism in these countries. Let's now look a little closer at the high fructose corn syrup consumption pattern in the U.S. through the years. In 2004, U.S. per capita intake of high fructose corn syrup was 60 pounds per year per person, according to the United States Department of Agriculture, USDA. Since there is no high fructose corn syrup consumption in the UK, we can conclude that the autism is lower in the UK between 2004 and 2010 time period compared to the US because of the difference in high fructose corn syrup consumption. No consumption in the UK versus between 50 and 60 pounds per year per US citizen. The mineral imbalances caused by high fructose corn syrup clearly creates conditions for autism, and the food colors laden with lead, mercury, and arsenic clearly create conditions for ADHD. Of course, pesticide exposures are also involved. With the pesticide exposures, we need our PON1 gene working correctly. What can modulate or suppress the PON1 gene in the U.S. to create more autism and ADHD? Environmental exposures to heavy metals such as mercury, lead, copper, and cadmium. Prenatal exposures to certain pharmaceutical drugs such as statins. Prenatal exposure to cigarette smoking, prenatal exposure to alcohol, and the dietary factors, mercury, lead, high fructose corn syrup, these exposures in particular. In the case of age, babies and the elderly have lower PON1 gene activity. The PON1 gene is part of the immune system with respect to the body's ability to detoxify pesticides. Boys and baby boys have lower levels of PON1 gene activity, thus autism and ADHD are more prevalent in males. Let's look a little harder at the dietary pesticide factor again. How is it we are exposed to so much pesticide in the U.S.? Where is some of this pesticide exposure coming from? In the U.S., conventional wheat grain may be stored in silos for up to 5 years. Malathion, a common organophosphate pesticide, may be applied to the grain as often as needed to kill the insects and their larvae. Malathion residues are always found in conventionally grown grains by the U.S. Department of Agriculture. These residues in wheat cannot exceed the allowable levels. So for 5 years, your conventional wheat may be stored in these silos and they can apply the malathion as needed to kill the larvae. In organic wheat, that wheat can only be stored for a year in a refrigerated silo, so they don't use pesticides. With organic wheat, the insect infestations are controlled through temperature. Colder temperatures prevent the larvae from hatching. So organophosphate pesticide exposures do in fact occur, and because the residues from these pesticides end up in the bread and other wheat and grain end products at low levels. Researchers continue to measure the amount of pesticide residue in wheat end products. The most recent study of pesticide residues in wheat end products was done in 2021. Wheat is the primary ingredient in breads and cereals. Children are high-end consumers of cereal and bread products with allowable levels of organophosphate pesticide residues. If a child's PON1 gene is not working properly, then she or he will have difficulty consuming these grain products. So just how much wheat do children in the U.S. eat? In our most recent research, we looked at what Americans eat on an annual basis and how what they eat might contribute to the development of autism and ADHD. We got our data from the USDA Food Availability Program. The most recent data available is for the year 2019. The data doesn't differentiate between child and adult consumption. As we can see, Americans eat a significant amount of wheat, 92.3 pounds per capita or per person in 2019. Americans have significantly increased their consumption of wheat. In 1970, they were only consuming 78 pounds per year per person. Compared to other farm products, wheat is the primary component of the Western diet. Wheat with allowable pesticide residues. Meanwhile, Americans are not eating enough calcium-rich foods. Calcium is needed by the PON1 gene to build the paraoxidase enzyme required by the body to break down the pesticides found in wheat. Milk consumption has declined significantly since 1970, when each American was consuming 179.5 pounds per year of milk. Now they're only consuming about 88 pounds per year. Milk consumption was at one point providing Americans with the dietary calcium they needed to stay healthy. Of course, cow's milk and dairy products are not the only source of calcium. Spinach and broccoli are also calcium-rich foods, but Americans are not eating much of those. 1.48 pounds of spinach and about 4.5 pounds of broccoli per person per year in 2019. Regardless of what we eat or drink today, the PON1 gene is not being supported by the American diet, which is no longer calcium-rich. The USDA does not track moringa consumption. It is worth noting, however, that an 8-ounce cup of moringa leaves provides 4 times the calcium provided by an 8-ounce glass of milk. Moringa is easy to grow, and we recommend growing it in your garden so your family will have an excellent source of calcium in their daily diet to support PON1 gene function. Another way to support PON1 gene function is to eliminate the sugar-sweetened beverages that contain high fructose corn syrup from your diet. As we can see, in 1970, Americans were consuming less than 1 pound per year of high fructose corn syrup, and in 2019, they were consuming 21.6 pounds per year per person. Remember, high fructose corn syrup suppresses the PON1 gene. Reviewing this slide one more time, in addition to high fructose corn syrup, what else can modulate or suppress the PON1 gene in the U.S. to create more autism and ADHD? Well, we already went over the environmental exposures to heavy metals such as mercury, lead, copper, cadmium, prenatal exposure to certain pharmaceutical drugs such as statins, prenatal exposure to cigarette smoke, prenatal exposure to alcohol, and the dietary factors of heavy metal residues, mercury, lead, and high fructose corn syrup as a separate exposure. In addition, high fructose corn syrup is a double agent because it is harmful in and of itself, and it is also a known carrier of mercury. And remember, baby boys have lower PON1 gene levels. So age and sex are a factor in the development of autism and ADHD. In 2010, Canadian government researchers duplicated our finding of mercury in food products containing high fructose corn syrup that were being sold in Canada. These researchers reported finding levels of mercury 10 times higher than the levels previously found by yet another group, the Institute for Agriculture and Trade Policy. The Canadian findings were published in the Environmental Health Journal as a comment under the paper we published in 2009. These studies indicate the average person is being exposed to inorganic mercury when they eat processed foods containing high fructose corn syrup. We are coming back to this slide. We've talked at length about heavy metal and pesticide exposures, how they occur. Let's focus on obesity and type 2 diabetes. These are two important disease conditions women need to avoid to reduce their risk of having a child wired to be autistic or to be diagnosed with ADHD. We stumbled on a finding of great importance while conducting a clinical trial at Fort Peck, Montana. We tested the hypothesis that inorganic mercury exposure comes from processed food. We administered 2 different interventions at the Fort Peck Community College over 10 weeks. In one intervention, we delivered an online nutrition intervention course. We tracked the dietary changes made by the students who completed the course. In another intervention, different students participated in a support group to eliminate corn sweeteners from their diet. We determined the changes in student health status after administering both of the 10-week interventions. The students in the online nutrition course significantly reduced their consumption of processed foods and increased their intake of whole foods by the end of the intervention. The students in the online nutrition course who reduced intake of all processed foods and increased their intake of whole foods had lower inorganic mercury levels in their blood compared to students who only eliminated high fructose corn syrup and other corn sweeteners from their diet. The one student who did not eliminate any processed food at all had the highest inorganic mercury level of all. In addition to the lower inorganic mercury levels, students in the online nutrition course had significantly lower fasting glucose levels at the end of the trial. Glucose is sugar levels, fasting sugar. So what we now know is that inorganic mercury residues are in many processed foods, not just those containing corn sweeteners. Humans are exposed to inorganic mercury when they eat highly processed foods. During this trial, we also stumbled on the finding that inorganic blood mercury levels appeared to be correlated with fasting glucose levels. To confirm this finding, we analyzed the CDC's NHANES dataset and found that inorganic mercury levels in the blood of 16,232 Americans are associated with fasting glucose or sugar levels. So the higher the inorganic mercury levels in the blood go, the higher the fasting glucose levels. Higher glucose levels are a risk factor for gestational diabetes and poor birth outcomes in pregnant women. Were there any changes in the health status of the 4 participants who were assigned to the corn sweetener elimination group? This table shows that there is significant difference between the average weight and BMI measurements that were taken during the study period. The participants who eliminated corn sweeteners from their diet showed significant losses in both weight and BMI. So here we can see that high fructose corn syrup consumption is clearly linked to obesity, which is a factor associated with giving birth to a child wired for autism or ADHD. This table also provides the fasting glucose data that show that there is a downward trend in the average fasting glucose between the intervention time periods. So when you eliminate corn sweeteners from your diet, you can expect that perhaps your glucose levels, your sugar levels will decrease. So now we know that heavy metal exposures from processed food intake are related to heavy metal levels in your blood. Kahn et al. Found heavy metal concentrations in foodstuffs eaten by adults and children matched the heavy metals found in their bloodstream. And of course, we found blood inorganic mercury is directly associated with glucose levels in the human population and may be linked to processed food intake. We also know that heavy metals in the blood correlate to the severity of social and/or cognitive impairment in children with autism and ADHD. Children with autism have higher levels of mercury and lead in their blood compared to healthy children. The higher the heavy metal levels, the more severe their impairment. That study was done by Al-Abdali et al. Wu found that processed food intake increases exposures to mercury and lead and is correlated to increased risk of ADHD. Another study, 2019, showed that the consumption of sugar additive, pesticides, GMO processed foods, and hard to digest starches may aggravate symptoms in children with autism. And of course, in our newest review article that is published in a methodology journal, evidence-based, we now know that autism and ADHD diagnoses and treatment may include the measurement of selenium, mercury, and lead levels in red blood cells and behavioral checklist data before and after healthy diet interventions. We can change the way autism and ADHD are diagnosed, and we can change child health outcomes with healthy dietary interventions. This is what the evidence is telling us. Clinical trial data from all over the world. We are back at the beginning of this slideshow with our newest model, which can be used by the medical community to diagnose and treat autism and ADHD in a way that will enhance child learning, behavior, and health outcomes. Children will have a fighting chance to succeed in school, and with the dietary changes, they will live healthier lives. They will suffer less frequently from type 2 diabetes, heart disease, Alzheimer's, and all of the other diseases that occur when we accumulate heavy metals and pesticides in our blood and body tissues. We're coming back to this slide. We can clearly see how diet impacts child health, behavior, and learning outcomes. We've looked at maternal obesity with high fructose corn syrup consumption, gestational type 2 diabetes with the processed foods, with the heavy metals, and the pesticide exposures with low PON1 gene activity. What can we do to educate prospective parents on what it means to eat a healthy diet from an epigenetics viewpoint? We can refer them to our healthy diet tutorial at the Food Ingredient and Health Research Institute. We can refer registered dietitians and other nutrition professionals to our free healthy diet tutorial at the Food Ingredient and Health Research Institute. The content has been successfully tested in 3 clinical trials targeting college students or parents of learning disabled children with autism and ADHD. Parents who participated in our online nutrition tutorial recommended the tutorial. When asked the question, would you recommend this tutorial to other parents, friends, or relatives, overwhelmingly answered yes. After participating in a 6-week healthy diet tutorial, our parents not only switched their families to a healthier diet, but they also saw improvements in their child's behavior. What makes our education tools effective? 8 modules of instruction, 1 for each chapter in the textbook. Each module provides learning goals, reading assignments, and discussion questions, 2 activities to meet the needs of different learning styles. We've got kitchen cupboard survey exercises, food ingredient label reading exercises, menu planning, video watching. It can be set up. Going through our tutorial can be set up as in a book study group. Upon completion of the tutorial, participants may take a 30-question quiz to qualify for the Food Ingredient Safety Specialist exam, now offered through Food Ingredient Health Research Institute. We need a whole army of food ingredient safety specialists to educate our population about the heavy metals in the food supply, where they're located, and how they impact our genes in conjunction with other factors such as the consumption of high fructose corn syrup. The textbook we are using is unique as it goes with our free online healthy diet tutorial, and it's written from the epigenetics point of view by a leading epigeneticist. This tutorial can be built on other organizations' websites. We're happy to help do that if anybody wants to. We're not going to charge any money. We just want to get people educated. Colleges already use the textbook, as do organizations who provide training in CEUs or CPEs for nutritionists and diabetes educators. All royalties from the textbook have been donated to the Food, Ingredient, and Health Research Institute. We can and will be unsafe at any meal if we do not understand how to avoid exposures to the heavy metal and pesticide residues allowed in the food supply by international and state regulators. Because the textbook is science-based and connects the reader to the learning objectives in the online tutorial, it plays an influential role in creating the interaction between the parents and their environments. During each of the 8 modules of instruction, participants are required to use the tables in the textbook to survey their kitchen cupboards to identify food products likely to contain harmful food ingredients or specify toxic substances. In using the textbook in this way, it serves as a tool of reciprocal determinism because it facilitates the interaction between the parent and her kitchen environment. The textbook provides the participants, the parents, with the means to effectively survey their kitchen cupboards and refrigerators to identify harmful food ingredients and highly processed foods as they learn to read food ingredient labels. The textbook is dedicated to my children, grandchildren, and the next generation. We want to acknowledge all those who have helped us get to where we are now to build the foundational science that we now have. We know what we know now because the United States EPA, Environmental Protection Agency, provided the funding for the 2 clinical trials at the Fort Peck Indian Reservation. The American Indian Higher Education Consortium provided administrative and moral support. The Fort Peck Community College and their students participated in the trials. The Food Ingredient and Health Research Institute hosted the tutorials on their website, and my friends and family funded the parent nutrition tutorial. FERI, donors, and volunteer science helped with all the publications. You know who you are, and today we give thanks to all of you. Have a great day. Be sure to eat healthy. And check out our website, www.foodingredient.info.